Welcome back everyone. In today's video, I will be discussing how you could apply your understanding of genetics and meiosis and genotype and phenotype to different situations where you can potentially predict the likelihood of somebody inheriting a certain trait based on their parents' phenotypes and genotypes. Before we start, I just need to give you a few more terminologies. The first new term is the word cross, which in this context means the breeding of two organisms to produce offspring. So you may see terms like a test cross or monohybrid cross. That's just basically breeding two organisms together and then observing the phenotype of the offspring. This sounds really strange that, I mean, obviously you can't really do a cross between two human beings just for the sake of an experiment, but it is actually done quite often in plants and animals, so in agriculture, just so that we can get a certain trait. So we'll talk about selective breeding maybe next week, but that's what a cross is. Pure breeding is when an organism is homozygous for that trait. So the organism has two copies of the same alleles of that trait. So for example, if somebody is AA or AA, so that sounds the same, but it's like all capital A's or all lowercase a. So that's pure breeding. Alternatively, if an organism is heterozygous for a trait, then you can say that it is a hybrid. I think these two terms you definitely have seen in other contexts. I think you could guess what they mean, but this is just to formally define them. So if an organism has two different copies of two different alleles of that trait, is a hybrid. A carrier, so a carrier is an individual who is heterozygous of a gene, so carries the recessive allele without expressing it. So for example, if there is a trait for black hair, and then there's a trait for red hair, so let's say this is black, and then, sorry, there's an allele for black hair and the other allele is for red hair, then a person who is heterozygous like this person's a carrier for the red hair trait, but the hair is still black. So a carrier is when you carry the allele for the trait, but the trait is not expressed. Okay, the P generation is the parents, and then F1 is the offspring of the parent, and F2 is the offspring of the offspring from interbreeding. So once again, this doesn't happen in human, but in agriculture, when we talk about plants and animals, interbreeding does occur. So you get from F1 to F2 generations. So those terms, I think we're going to use them, and then you don't have to memorize these. I think they're pretty self-explanatory. Anyway, so let's start with the first example. I think I have to put in a disclaimer that the development of dimples come from multiple sources. It's not just a genetic condition. So just because your parents don't have dimples doesn't necessarily mean that you will not have it. So it can be developed during the development of the fetus and things like that. And the environment does play a role in how the trait is going to be expressed as well. But just for the sake of simplicity, we're going to assume that dimple is a genetic trait. So the dimple allele is dominant. So we're going to use capital letter D to represent it. And the recessive allele is no dimple, and we're going to use the lowercase d to represent it. So the question is this: if a man has dimples, and I actually go one step further and give you the genotype of the man here. So if a man with dimples with this particular genotype and a woman with no dimples with this particular genotype, if they're having a baby, what is the probability that the child will have dimples? I don't think anyone would care that much, but let's just assume we do. So how can we use our understanding of genetics and meiosis to answer this question? And obviously in a real life context, the condition that you're probably interested in is not dimples, but maybe something else. And we'll look at some of those contexts later. So let's just go back to the process of fertilization. So let's say this is the female, so this is the woman, and the woman contains 46 chromosomes in any normal cell, and the woman in this case has a genotype small d. So this is the woman. So via meiosis, this cell is going to be split into 4 cells, 4 gametes, and each gamete is going to contain one copy of the lowercase d allele. So you basically just have 4 of these, but they're identical. So you basically have 2 different types of egg cells that could be formed. One type of egg cell that could be formed. So you just have this. but there are 4 of them. Let me just draw it here. So basically, via meiosis, because if you remember, meiosis is a process that half the amount of chromosomes that a cell has. So via meiosis, you're going to have 4 gametes from the female, and each gamete is going to contain 1 allele, 1 lowercase d. Now, if you look at the man, the genotype of the man is lowercase d and capital D. So via meiosis, you're basically going to have 2 cells that can have the capital D allele and 2 cells that can have the lowercase d allele. So this is why we need to understand meiosis, because meiosis explains how you can have these. Okay, and then one of these cells, it's going to fuse with one of these cells to produce a child, and you have to predict what is the probability that the child is going to have dimples. So it really depends on the possible genotype that the child can have. So I think in this case, you look at this, so there's really only one type of gamete as far as the D genotype is concerned. There's only one type of allele that the mother can give the child, which is lowercase d. And there are 2 different types of alleles that the father can give the child. So the child can have 2 possible alleles, either capital D lowercase d or lowercase d lowercase d. So in the first case, the child will have dimples, and in the second case, no dimples. And the probability here is going to be an even split, so 50%, there's a 50% chance that the child will have dimples and 50% chance that the child will not, because 50% of the gametes from the father has the allele for dimples and 50% doesn't. So that's how you would do this question. However, in the field of genetics, we normally use another system just to represent all this information without having to draw this entire diagram and then draw the 4 cells coming out of meiosis. And that system is called the Punnett Square or the Punnett grid. Or I think we're going to stick with Punnett Square, but I've seen it being called something else in other contexts. So just Punnett something or other. And it is essentially a structure, a table, if you will, that allows us to predict the likelihood of offspring inheriting a specific genotype or phenotype. So the possible combinations of alleles, I'll go through what this means. The possible combinations of alleles that the parents contribute are placed at the top and left-hand side of the Punnett square, and then the possible combination of alleles of the offspring that the offspring could have are placed in the blank spaces. And then from there, you can predict what is called the genotypic ratio and phenotypic ratio of the offspring. So I'm going to give you some steps that you can follow whenever you do an inheritance question using the Punnett square. First one is to I'll go designate letters to represent the alleles. I was going to say I'm going to go through them with the examples, but let's just quickly run through them. And then you can write down the phenotype, the genotype of the prospective parents, which is your P generation. And then you write out the genotype of the parent gametes and draw the Punnett square, and then you can complete it. And then from there, you should be able to determine the potential genotypes and phenotype of the offspring, which is in this case the F1 generation. It may look like a lot, but it really isn't. Like the first 4 steps can be done within the table. Okay, so that's my animation is not great there, but let's just go back to example 1. I'm going to use the Punnett square method to solve this question, so I don't have to go through the whole gamut. So step 1 is to designate letters represent the alleles. This has already been done for us, so we already know that capital D represent the dimple alleles and lowercase d represent the no dimple alleles. If this is not given to you, then you have to start with that. Step 2 is to write out the phenotype and genotype of the prospective parents. This also has been done for you in this question. I know that the father, his phenotype that he has dimples and his genotype is heterozygous at that trait, and the mother, her phenotype that she has no dimples and her genotype is that she is homozygous recessive at that trait. So I already have step 2 done for me by the question. Step 3 is to write out the genotype of the parental gametes. So step 3, this is where meiosis comes in. But if the father has genotype capital D, and lowercase d, it means that there are two possible gamete genotypes that he can give the child, which is capital D and lowercase d. So the father could give either one of these, not both, but either one of these allele to the child, and the mother can also, because her genotype is just lowercase d, she really can only give one type of allele to the child. And then from there, we're going to draw our Punnett square. I think from the next step, I'm going to do step 3 and 4 at the same time. So the way that the Punnett square works is you just draw a table, and it really doesn't matter where you put the mother and the father, but we're going to do a table like this. So this is a square. Some people care a lot about, straight and lines being straight, but quite frankly, I really do not care. As long as you draw a decent table, that's okay. So the father gametes I'm going to put on top, but again, oh yeah, okay, so we want the maternal gametes along the top. All right, we can do that. So let's say this is the mother. So there are two possible, well, there's really only one possible gamete that she can produce as far as this trait is concerned, but we're just going to put each allele that she can give the child into each column. So this is from the mother, and then the father is going to be this part. So the father's alleles will be here. So we're just going to go capital D and lowercase d. So once you get very good, you can just start with the Punnett square. Okay, and then the last step is to actually complete the Punnett square to determine the genotype of the offspring. So the genotype of the offspring is just a combination of one allele from the father and one allele from the mother. So it could be capital D, lowercase d, lowercase d, lowercase d, and then this again. So if you look at this, you should be able to work out. I'm just going to write father. Oh, sorry about that. Just going to move this up here so I have a little bit more space to write the final. Okay, so from here you should be able to work out what is called the genotypic ratio. So genotypic ratio is 50%, or 2 out of 4 of the possible allele combinations of the child is capital D, lowercase d. So the genotypic ratio is this, capital D, lowercase d. This is a 1-to-1 ratio, or so 2 by 2 if you will. So there's 2 of one and 2 of the other, so 2 over 2, which you can simplify to a 1-by-1 ratio. Now the phenotypic ratio is when you need to be able to translate from a genotype down back to a phenotype. So if somebody has a genotype of capital D, lowercase d, they have dimples. So dimples there is no dimple. This is also going to be a 2 by 2 or 1 by 1 ratio, because 50% of these possible allele combinations is with dimples and the others is without dimple. And if you answer the question, what is the probability that the child will have dimples? The probability is 50%. There is a 50% chance that the child will have dimples, but I ran out of room to write that. So I may just write it up here. So what is the probability? There is a 50% chance that the child will have dimples. Okay, so that's the first example of Punnett squares.